Welcome back, everybody. This is registered dietitian Myrna Haag, where I bring you, hopefully, very impactful nutrition information you can apply to your life and really benefit your health. So what we're going to talk about in this video is some gene testing. And these are particularly the gene tests that I do. And I'm only going to explain one in each video because I think they each deserve their own attention. So what we're going to talk about in this video is the APOE gene. And Sam, if you're ready, let's flip on to that PowerPoint. All right, awesome. So the top gene tests that I think doctors should be doing, and you can pretty much ask your doctor about the APOE gene, and Sammy, will I be able to switch to the, okay, perfect. So what is nutrition, a medical nutrition therapy? What exactly is that? Well, that's what registered dietitians are licensed to do. And it basically takes into account your genetic profile, your laboratory values, anything clinically that we can get our hands on. We're going to take that information and we're going to be able to give you a diet that's either going to help you benefit maybe a diagnosis you have from a biomarker, laboratory value, or in this case, a particular gene. So we're going to talk about the genes from the reference of, well, where are genes really? What are they? They're found in the DNA, and they're a group of protein sequences that have instructions. So each gene has a code of instructions to perform some kind of function. So think of genes as sort of like an instructional tablet, right? It goes through the DNA and it pretty much tells these proteins where they're supposed to go and what exactly they're supposed to do. And some of the genes that I look to do in my practice, the specialty genes for my medical nutrition therapy, is the APOE, which is the one we're going to talk about in this video, the APOB, which we'll have to address in another video, lipo(a), the MTHFR gene, which I've already done a video on it, we'll bring that up, and then the heptoglobin 2. That's a really interesting gene, and interesting why I would look at that gene. And then LDLP and LDL small. So those are the size and the particle number of your LDL. And those do have a genetic reference there. And they're important, and that's important to get that information. So let's dive right in to the APOE gene. So the APO gene is a principal fat carrier for the brain. Okay, and a person with the APO4 variant will have a greater risk for cardiovascular disease and Alzheimer's. So let's talk about, before we get into the different variants, the different types of gene properties. We have what are called epigenetics and actual gene mutations. So when you think of genes, these are these proteins that are sequenced to do a certain thing, instructions that are coming from that DNA. You have what are called epigenetics, and this is reversible. And this is when an environment or some kind of toxin or something in the environment or a diet change, or something in the environment or a diet, and how that can change how the gene responds in the body. So it's almost like these genes are going to be affected by the environment, by your diet, and go to another body part and cause some type of destruction. So the gene actually works fine. So there's no problem with the gene, but it's what's going on in the environment, maybe a toxin, or maybe what's going on in the diet that affects how these genes will react in a certain area of the body. So these genes have instructions to do what they do, and when they're doing what they do at certain specific areas in the body, depending on your diet or maybe something in the environment, it affects the function. okay? So epigenetics means the gene's fine, but the function's not doing so well because of something in the diet or maybe something in the environment. A gene mutation is a little different. That's not reversible. Oh, by the way, the reason why epigenetics is reversible is you just adjust whatever compound is in the environment or whatever is in the diet, and you can change how that gene will respond. So that's kind of great, right? Because that's good news. So the gene's not a problem. We just have to fix what's in the environment and the diet. Gene mutation, that's different. That's not reversible. That's irreversible. And that's going to actually be a change in the messaging, the actual sequencing of this gene, because it's a bunch of protein sequencing is not sequenced properly. Somewhere along the line, one of those proteins got damaged, got altered. But unfortunately, what that means is the gene no longer is sending the message that it should send. So that's a mutation. It's an alteration in the gene. So the ones that we're going to talk about, and the ones as a dietitian are so important, are the ones that are epigenetic related. By the way, gene mutations, a lot of times you can have a gene, and it doesn't mean that you might have a certain type of gene for a certain disease, but it doesn't necessarily mean you're going to get the mutation. That can happen. Sometimes certain things can trigger it. So you can have a gene, doesn't mean it's necessarily going to be expressed. I just want to throw that out there. Okay, so let's talk about the epigenetics and what exactly that means, a little more detail. It refers to the way your behaviors and environment can cause changes that affect the way your genes work. Epigenetics turn genes on or off. They basically have genes doing things maybe differently in a certain area. Your epigenetics change as you age, both as part of a normal development and aging, and because of exposure to environmental factors that happen over the course of your life. Epigenetic changes can affect your health in different ways. So certain things in your body can do that as well, just like they said, aging. And there's certain factors in aging which we can talk about, I think, that are interesting. We should do a podcast just on aging and some epigenetics that goes on there. But so certain things happen to you that affect how some of these genes are going to function. So one of them, and I want to give a good example, is obesity. So obesity has to do with excess fat cells that grow, excess number, excess size, and it causes an increase in two inflammatory immune cells, two inflammatory compounds.
Zoom out of the PowerPoint a bit.
Okay, zoom out.
Yeah, your picture was blocking.
Okay, perfect. And this causes an increase in these compounds called interleukin-6, TNF, tumor necrosis factor. And what happens is when you have a lot of release of these excess fatty acids, because either you're too heavy, you got too much excess fat, a lot of times it can even be because you're eating too many dietary fats, which I talk about that a lot in my diabetes videos. But this can cause an accumulation in cells. And with the APOE, we're going to show how this can be a real problem in the brain and in the vascular system. So with obesity, that would be an environmental thing. That would be something that's happening to you because you were taking in maybe too much food, you were getting fatter. Excess fats could also do this, increasing the amount of fatty acids in your blood. And basically, this triggers these immune cells to come into play. And what this does is it causes genes to function differently. And we'll take into account type 2 diabetes, and we'll show the insulin gene, the genes that make insulin, and how these excess fatty acids can affect it. So you have the gene that's making insulin. Insulin is going to the receptor site on the cell, particularly muscle cells, because that's going to be the cells that have these insulin receptor sites. Also brain cells do as well. And what happens is insulin is the hormone that attaches to these receptor sites, and it allows a messenger to take place in the cell that then brings up a transporter that then lets glucose inside the cell. Well, what can happen is that you can have a lot of tumor necrosis factor, you can have a lot of excess fatty acids clogging up this receptor site, where now insulin is not able to do its genetic function, which is to allow sugar into the muscle cell. So here in epigenetics, we have an example how obesity, excess fatty acids that are accumulating in this muscle, or these excess immune TNF, interleukin 6, these immune cells can clog up this receptor site, okay, causing this epigenetics where now insulin is not able to function like it should. So that's an example of the compounds of these immune cells, interleukin 6 and TNF and excess fatty acids, have affected how insulin can now not function like it should. So when we now look at the APOE4, and with the APOE4, this is a gene. Let's just go back to the definition, so before we get into some of the studies, we're really clear as to what the gene does and what its purpose is. This APOE is a fat carrier, so it carries cholesterol and fats through the blood. And the APO4 has different gene variants, so the APO4 is a carrier. And let me see if I can find the right slide here, because I think I might have had them out of order. Let me see. Okay, no, I'm looking for the right slide here. I thought I had it in the order, so. Okay. Nope, can't find it. That's all right. I might not have included it. So the APO4, if we look at this study, it's linked to damaging lipid droplets in Alzheimer's disease brain cells. So several genetic risk factors for Alzheimer's disease is implicated in genes involved in lipid metabolism. So the key with the APO4 gene is it actually helps carry cholesterols and fats through the blood. And in the brain has these fat storage in certain neuronal cells in the brain. And when you have this APO4 genotype, it is actually affected. So the carrier there with this epigenetics with the APOE4, here it is. Okay, nope, I can't seem to find it. I must have forgotten that slide. But with the APO4, the real connection there is that it has it is affected, the epigenetics in this is affected with excess fatty acids. So the excess fatty acids, these fats that are circulating in the blood and also in the brain, are affected with this particular gene type. So the relationship between, in this study, lipid metabolism in the glial cells in Alzheimer's disease pathology remains poorly understood. So this shows in the green highlighted area, we have identified the microglia state, which defines the expression of these lipid droplets associated with an enzyme of ACSL1. So the point here is that these ACLS1 enzymes that help with these lipid droplets are abundant in patients with the Alzheimer's disease having the APO44 genotype. So the APO44 genotype is implicated with these enzymes that are used with these lipid droplets in helping clear them from the brain. But the APO44 genotype, unfortunately, does not have good clearance, possibly due because of this enzyme that doesn't work as well for the APO44. Now, the APOE genotypes have 3 genotypes. Let me see. I thought I did that slide. Let me see if I can find it so I can show you the different genotypes. Let me see.
Do you mean APOE and all those?
Yeah.
That slide's not there.
Oh, it's not? Okay. This shows the different variants?
I can put it in.
Okay, that would be great. So Sammy's going to put it in for me, and we will take you through the different variants. We have the variant 2, 3.
You're going to have to talk about something. It's going to take me a minute.
Okay. We have the variant 2. Well, I can just pull it up here because I already know it. I don't really need the slide. I can write it. So we have the APOE 2, 3, and 4. Now, when you have genes, you have 2 chromosomes. You have 2 alleles that make up these chromosomes, one from your mom and one from your dad. So the variants are either a 2-2, 2-3, 3-3, you got a 3-4, and you've got a 4-4. And so remember, with these alleles, you got one from your mom, one from your dad. So with these chromosomes, when you have the genotype 4-4, this is the genotype that has a lot of these problems with the lipid droplets. This is a genotype that we're seeing doesn't do well with this enzyme doesn't function well that helps clear these lipids from the brain. So when you're a 2-2, 2-3, 3-3, or possibly a 3-4, when you become a 4-4, you have up to 60% chance of getting Alzheimer's by the time you're 80, okay? And it starts as early as in your 60s when you're the genotype 4-4. You have a 60% chance. So, Sam, if you can find those slides, because I know
I can't. It's going to be too long.
Okay, no problem. So if you, and I got that from the CDC, and one of the slides showed that the CDC, Center for Disease Control, talked about with the 4-4 genotype that there was this percentage risk that they would develop Alzheimer's. So what that means is that when you have this 4-4 gene, you're going to be at greater risk, okay, the 4-4 gene. And this is what they're referring to in these studies, these 4-4 genotype. So when you go to your doctor and you tell your doctor, I want to get the gene testing for the APOE gene, you're looking to see what variant you are. Are you a 2-2, 2-3, 3-4, or a 4-4? It's that 4 genotype. So this is what these studies show, is that our findings suggest there's a link between the genetic risk for Alzheimer's disease and these microglia lipid droplet accumulation that causes this neurotoxic problem in the brain. And this is what's contributing for these APO4s Alzheimer's risk. So if we go to this particular study, it talks about Alzheimer's original description of what would later be called Alzheimer's disease included the definition, identification of these glia cells. And it shows the adipose saccules, which is these little fat saccules, the little fat vesicles in the brain of patients with dementia. And so this is what contributes to the risk factor for Alzheimer's. So the APOE is one of these risk factors, 4-4, and it's that variant in some of these mouse models that they found that these accumulation of the lipid droplets with the 4-4 is what triggered a lot of the problem. So I wanted to talk about one particular study by some doctors, and I thought this was a great example, and I wanted to read it to you, but protein accumulation on fat droplets implicated in late onset Alzheimer's disease. In this particular study, Chapel Hill, North Carolina, UNC School of Medicine researcher Sarah Cohen and Ian Winham, a former PhD student from Cohen Lab, have made a new discovery about the APOE gene, the biggest genetic risk factor for late onset Alzheimer's disease. And in the green highlighted, I show here, it's a 5-year-long study to better understand and visualize the relationship between the APO4 and Alzheimer's disease and fat molecules in the brain. We discovered that brain cells known as astrocytes are more vulnerable to damage and may even go dysfunctional when APOE4 surrounds these lipid storage centers. So in the brain, you've got these little fat storage centers, and according to Dr. Cohen, that these she authored a paper on this and how this exactly works with the APO4 genotypes in Alzheimer's disease. So let's go a little deeper into some of her work. The role of lipids in the brain. 60% of the brain dry mass is composed of lipids, which play an important role in the brain, such as storing cellular energy and forming myelin sheaths, the substance that surrounds and insulates those neurons. Lipids can be found in specialized fat storage compartments, so lipids being fat, known as lipid droplets within the astrocytes, and those are brain cells. So within the brain cells, we have these little lipid or fat storage containers. Astrocytes are tasked with cleaning up the free-floating toxic lipids. And what happens is if you're an APO4 genotype, your astrocytes do not do a good job in cleaning up those lipid droplets. So the astrocytes were becoming damaged, dysfunctional, in a way that they could not perform their cleaning duties. And this was particularly found in the APO4. And this talks in her study, the APO is produced by the astrocytes, much like a taxi or Uber. The protein oversees releasing the transporting of these lipids. And what they found when they did their study using a microscope, that the team observed an usual formulation of these APO4s, but like they normally would go, but these particular APO4 genotypes, they caused problems. They were actually contributing to more of the plaque formation that causes Alzheimer's, the amyloid beta plaques. So they thought this was a very interesting result to the study, but it has already been found in several journals looking at these APO4 genotypes and Alzheimer's, and not only in Alzheimer's, but the APO4 is also implicated in cardiovascular disease, because these APO4 genotypes, they actually do not produce a lot of clearing of the bad cholesterols as well. So they don't do it in the brain with some of these fat droplets, and they're not as good at clearing the LDLs, and they actually increase the LDL levels because of that. So the APO4s basically means that you're not going to be good clearers of fat out of the brain. So what would be my recommendation? Because we know that these excess fats, where are they coming from? Well, being obese is one area, and also excess dietary fats. So eating too many dietary fats or just being really obese can affect these excess fatty acids. And for the APO4s, this is not going to be a good situation. So let's talk about how we fix the situation, because that's super important. And what the recommendation is, we want to reduce our fats less than 25%. And I want to show you how to get to that figure, because a lot of people say, well, great, what does that mean? I'm going to give you a number in this video so that you have a reference. But definitely, if you're an APO4, you want to avoid the keto diet or any high-fat diets, because it is toxic for APO4/4s, which now you know why. As a dietitian, I definitely want to get that genotype, the 4/4. I want to see if my patients have that gene, because epigenetics tells me if I can reduce the fats out of their diet, then I can reduce the risk. Another good recommendation is to have the DHA and the EPA. These are the good fats, the omega-3. So ideally, I like to see 2 grams of the DHA, and I just kind of did a supplement label here so you could get an idea. And let me see if I can move that. There we go. Oops, let me see. There we go. So in this particular supplement, it shows DHA, which is the active one, the one we're really looking at. We want to try to get 2 grams. So this is almost a gram at 790. So that's 2 soft gels. So I would probably say you would need 4 soft gels here, maybe even 4 to 5, just to give you an idea. So that's the recommendation for, because we want to keep the healthy fats, the good fats, the omega-3 fats that actually help with the brain. But, I know the keto diet's out there a lot. Everybody's doing it. One of my podcasts, I did kind of mention some of the positive things with the keto diet, but I'm not a real advocate of it. I realize that there can be some benefits to it, but overall, I don't see it as advantageous as managing the macronutrients and getting the protein, fiber, fats correct, and the carbs. Getting those macronutrients at the right percentages is really the best bang for your buck as far as your health and also as far as your weight. So let's look at how we get to a 25% fat. How do you know, looking at fat grams, what that should be? And if we look at a food label, we're going to see fat grams on there. So how this is done is we're going to look at first how we get to our ideal body weight. Because if you're going to try to figure out macronutrients for fats, it's based on taking your ideal body weight and dividing it by 3, and that's going to give you 25% in grams. Because to tell you 25% of your diet you need or less of fat, if you're an APO44, doesn't help you a doggone bit, because you're looking at your plate of food and you're like, what does that mean? To tell you, just do a low-fat diet also doesn't give you some type of benchmark. So how do we get to that number? How do I get to that fat gram number? Because I can look up fat grams in food. For instance, 1 tablespoon of olive oil is 15 grams of fat. 1 avocado is 20 grams of fat. So you can look at every label and at least get an idea of what the value in fat is in your foods. So how do we get that number for you? Well, first thing is we got to figure out what's your ideal body weight. Now, some people may already know it. Some people may not. So I want to give you a formula that gets you there. And here's the formula. For women, anything over, we're going to take 100 pounds as our base, and every inch over 5 feet, we are going to add 5 pounds and add the 100. So if my height is 5'2", and I take away 5 feet, so for every inch over 5 feet, we're going to add 5 pounds. So if I'm 5'2", that means 2 inches. I take 2 inches times 5, because it's 5 pounds for every inch over 5 feet, and that's going to give me 10, and I add it to 100 pounds, so my ideal body weight is 110. Now, that's going to give you a BMI of like 22. And that's probably right in the middle of where you should be, because we want a BMI of anywhere between 19 and 25. Okay, it's really 18.9 and 24.9. But this is going to be smack in the middle. So you could add 10 pounds and still be within your correct ideal body weight. So it could be 110 plus another 10 pounds. So really 110 to 120 pounds would be a good ideal body weight for somebody who's 5'2". For a male, we're going to take, for 5 feet, every inch over 5 feet, 6 pounds and add it to 106. So if a male is 5'10", and I take away 5 feet, I come up with 10 inches, and 10 times 6 is 60, plus 106. I've got 166 as an ideal body weight. I can add 10 pounds. So we have got an ideal body weight of 166 to 176. Any one of these numbers is fine, still within your ideal body weight. So if we go back to figuring out what is it for me with this 25, because I want to, if I'm an APO44, I want to try to keep my fats under 25. So if I'm 166, let me get my phone and do the math on that. And I'm going to give it to you right away. Oops, there we go. So if I divide that by 3, that's 55. So I have 55 grams of fat for 25% of my daily value. So at least that gives you a number. And if you're 120 pounds, you do the same thing. You divide it by 3, and that's going to give you 25% or less of your daily fat value. And most of my patients, I try to put them at 20%. That's a pretty good number because, it's really tough to, because your brain does need fats. It's those omega-3 fats that are so good for the brain. So 20%, anything under 25%, most cardiologists are going to tell you you're within the green zone. You're good. It's going to help lower that toxicity that some of these fats can cause. So if we go back to what are the recommendations, we want to keep our dietary fats less than 25%. And for most of my patients, I have them like 20%, 20 to 25. And we want to avoid the keto diet, bad diet if you're an APO44. And we want to bring in those omega-3s. 1 to 2 grams, preferably I'm putting my patients with the APO44 2 grams. So that pretty much covers it. In review, the APO4 is a gene that most doctors should know about. Does insurance cover it? Some providers do, some don't. But you can most doctors can get this for you. I highly recommend it. And if you are an APO4, especially if you're a 4-4, if you're an APO3-4, I would still keep my dietary fats within 25 to 30%. And as a dietitian, I don't recommend anybody to have their dietary fats more than 35%, 25 to 35. You're an APO44, I'm going to put them a little lower, right? So there's a reason why the macronutrient percentages are the way they are as far as what we need in a day. And the dietary fats, for me, as a dietitian, it's important to understand that excess of anything isn't good. And some of these diets that have an excess of fats, especially with certain gene types, with the APO4 being one of them, can be a real problem. Okay, so I wanted to leave it open for some questions. If any of you have questions, Sam, if you want to take it into the chat.
Yeah, just 1 second. Let me get that. Chat. There was one that I put in there.
Okay.
Do you see it?
Do you have any stories of a patient who had an APO gene? Yes, I have, and thanks for mentioning that. I have patients that when I do get their gene types, and they have the APO44, a lot of times they are on these keto diets or they're on these high-fat diets. And unfortunately, there's no real biomarker that I can go in and say, we're like I can with atherosclerosis. I can look at their lipid panel. I can see diet can have a change there. In particularly with the APO4, there's no way that I'm going to be able to know this low-fat diet is improving their health, right? Because unfortunately, with Alzheimer's, once you start getting the scarring, once you start getting the disease, that's usually the diagnosis, unless you get a brain scan. So many times, people don't really know they have it. And putting them on a low-fat diet, a lot of times I don't really have a biomarker. I just know that if they have the APO44 and with the studies and with the pathology and how that gene works, I know with epigenetics, I definitely want to be, have my patient be aware that these fat grams that they're taking in can be a real problem. I want them to be aware of that. So I don't know if that helps Sam with the question,
Yeah, there's another
but
one too.
awesome. So if we don't have any other questions,
There's another one.
let me see, bring it up.
Oh.
Could you do chia seeds or fish? Yes.
Read the whole thing.
Let me blow it up. Could you do chia seeds or fish for omega-3 instead of a supplement? Yeah, you really can. So again, you want to look at the amount. So 3 ounces of a salmon is going to give you, I think, about 1 to 2 grams of the omega-3. And 1 tablespoon of chia seeds, actually, I know 1 tablespoon of flax seeds, you're up really high. You're up around 8 or 9 grams, a lot of omega-3. And chia seeds may be a little lower. You're probably looking about 3 or 4 grams. So that's actually
So 1 ounce of its.
one.
I was gonna try to look it up.
Yeah, go look it up. 1 ounce of chia seeds.
Chia seeds, I think, is 2.53 grams of omega-3s.
Perfect. So that's 2.5. So that's great. And then you also have the look up the flaxseed. I know flaxseed is much higher. It's more concentrated. And if you do the flaxseed, you want to make sure that it's ground flaxseed, not the whole little flax seeds, because 1.8. So it's for 1 ounce.
For flax seeds,
Yeah.
contains 1.8 of plant omega-3s for each tablespoon.
Oh, tablespoon. Okay, great. What's the tablespoon of chia? Because the doesn't show, right?
That was
So
what I told you.
So chia, 1 ounce of chia may be
It says
probably.
a tablespoon of chia is 2.53.
Okay, so chia is more than the flax?
That's
Wow,
what
I
Google
thought the
says.
flax.
I'm like,
Google says it.
roughly
We'll have to,
searching.
yeah, we'll have to get back at that because I thought the flax was more than the chia.
It might be.
Okay. Well, we'll take Google's word for it, I guess. Okay. Any other questions?
I think that's it.
Okay, awesome. Thanks, guys. If you have any other topics that you want me to go over, I am going to go through in the podcasting all the gene sequences. This is just APOE4, but I will go through the rest of them in the podcast to come. So you get a really good idea what they do, how epigenetics affects them, because it's all about the epigenetics. I'm not going to be doing the gene mutation ones, right? Because once you get that, that's a little different. So join me. Let me run through for you so we can better your health. Like and subscribe. And is there anything I should be doing else?
No, that's
Okay,
fine.
great. All right, awesome. Okay, thanks, guys, and I'll see you in the next stream.